Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Brucella Agar: Composition, Principles, Preparation, Uses, and Limitations. Table of Contents. Introduction to Brucella Agar. Composition of Brucella Agar. Principle of Brucella Agar. Preparation and method of use of Brucella Agar. Result interpretation on Brucella Agar. Uses of Brucella Agar. Limitations of Brucella Agar. Introduction to Brucella Agar. Brucella is a notable intracellular parasite recognized for its capacity to cause significant health challenges, including epizootic abortions in animals and febrile illnesses or localized infections in humans. These pathogens are characterized by their fastidious nature, necessitating a nutrient-rich environment for robust growth. Handling Brucella demands stringent precautions due to its high infectivity, underscoring the importance of rigorous biosafety protocols in laboratory settings. Brucella agar base supplemented with appropriate nutrients is specifically formulated to facilitate the enrichment, isolation, and cultivation of Brucella and Campylobacter species from a diverse array of clinical and environmental specimens. This specialized medium not only supports the growth of Brucella, but also serves as a conducive substrate for other fastidious bacteria such as Streptococci, Pneumococci, Listeria, Neisseria, Meningitis, and Haemophilus influenzae. This adaptability makes Brucella agar an invaluable tool in microbiological laboratories for studying and diagnosing infections caused by these diverse bacterial pathogens. Its ability to foster the growth and isolation of such organisms underscores its pivotal role in microbiological research and clinical diagnostics. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Composition of Brucella agar. Ingredients: grams per liter. Enzymatic digestive casein, 10 grams per liter. Enzymatic digestive animal tissue, 10 grams per liter. Yeast extract, 2 grams per liter. Sodium chloride, 5 grams per liter. Dextrose, 1 grams per liter. Sodium bisulfite, 0.100 grams per liter. Agar, 15 grams per liter. Final pH, 7.0, 0.2 at 25 degrees C. Principle of Brucella agar. Brucella agar operates on a principle that harnesses a meticulously balanced composition to support the growth of fastidious microorganisms. This specialized medium contains essential components that cater to the nutritional needs of these organisms. Peptones are integral as they furnish organic nitrogen, which is crucial for microbial metabolism and growth. This nutrient ensures that microorganisms have the necessary building blocks for synthesizing proteins and other vital cellular components. Yeast extract plays a pivotal role by providing a rich source of B-complex vitamins. These vitamins are essential cofactors in numerous metabolic processes within microbial cells, facilitating enzymatic reactions that are fundamental for growth and replication. Dextrose serves as a primary energy source for microorganisms. It is readily metabolized through glycolysis, generating ATP, adenosine triphosphate, molecules that power cellular functions and drive growth processes. Sodium bisulfite acts as a reducing agent within the medium. By maintaining a reducing environment, it helps to preserve the integrity of sensitive compounds and enzymatic activities necessary for microbial viability and growth. Sodium chloride contributes to maintaining osmotic balance, crucial for microbial physiology. It ensures that cells maintain appropriate water balance and ion concentrations, which are essential for enzymatic reactions, nutrient transport, and overall cellular homeostasis. Agar, the solidifying agent in Brucella agar, provides a stable matrix that supports microbial growth and facilitates the isolation of individual colonies. Its gel-like consistency allows for the diffusion of nutrients and gases while providing a surface for microbial attachment and colony formation. Preparation and method of use of Brucella agar. Suspend 43 gram of the medium in 1 liter of purified water. Heat with frequent agitation and boil for 1 minute to ensure complete dissolution. Autoclave at 121 degrees Celsius for 15 minutes to sterilize. Pour the sterile medium into sterile Petri plates. Follow standard procedures to obtain isolated colonies from specimens. Incubate plates at 35 AS 2 degrees C for 72 hours in an aerobic atmosphere supplemented with carbon dioxide, as many pathogens require carbon dioxide for primary isolation. Result interpretation on Brucella Agar. After incubation, most plates typically exhibit confluent growth. The streaking procedure acts as a dilution technique, depositing decreasing numbers of microorganisms across streaked areas. As a result, some of these areas should display isolated colonies of the organisms present in the specimen. Furthermore, the growth of each organism can be semi-quantitatively assessed based on its growth in each streaked area. In the culture results on Brucella Agar, Brucella melitensis demonstrates robust growth characterized by transparent, raised convex colonies with smooth, shiny surfaces and entire edges. Similarly, Brucella suis also shows luxuriant growth, forming transparent, raised, convex colonies with smooth surfaces and entire edges. Brucella ovis exhibits good growth with convex colonies. Streptococcus pyogenes demonstrates vigorous growth on the medium. In contrast, Staphylococcus aureus sub-sp. Aureus and Escherichia coli both show inhibited growth under these conditions. These observations highlight the differential responses of various organisms to Brucella agar, emphasizing its utility in microbial culture and identification studies. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Uses of Brucella agar, used for cultivating Brucella spp and other fastidious microorganisms in laboratory settings. Functions as a general-purpose medium for cultivating Streptococcus pneumoniae, Streptococcus viridans, and Neisseria meningitatis. With the addition of blood, Brucella agar is employed to determine bacterial hemolytic reactions. Serves as a base medium for isolating Campylobacter. Recommended by APA for isolating Brucella species from food samples. With the addition of 5% horse blood, the medium is utilized in qualitative procedures for isolating and cultivating both non-fastidious and fastidious microorganisms from various clinical and non-clinical specimens. Quality control PF Brucella agar. In quality control testing conducted by anaerobe systems, several organisms are routinely used to ensure the reliability and effectiveness of anaerobic blood agars. These organisms, specified by the Clinical and Laboratory Standards Institute, CLSI, provide critical benchmarks for assessing the performance of the medium. Here are some key details. Bacteroides fragilis, ATCC 25285, shows growth within 24 hours of incubation. Prevotella melaninogenica, ATCC 25845, typically exhibits growth within 24 to 48 hours and may produce a distinctive tan-colored pigment. Fusobacterium necrophorum, ATCC 25286, demonstrates growth within 24 hours. Fusobacterium nucleatum, ATCC 25586, shows growth within 24 hours. Clostridium perfringens, ATCC 13124, known for displaying a double zone of B hemolysis after 24 hours of incubation. Peptostreptococcus anaerobius, ATCC 27337, exhibits growth within 24 hours. Staphylococcus aureus or Enterococcus faecalis, ATCC 25923 or ATCC 29212. Both organisms demonstrate growth within 24 hours. Escherichia coli, ATCC 25922, shows growth within 24 hours. Proteus mirabilis, ATCC 12453, displays growth within 24 hours. Propionibacterium acnes or Clostridium difficile, ATCC 6919 or ATCC 9689, typically shows growth within 24 to 48 hours. It's important to note that pigment production by Prevotella melaninogenica may require more than 48 hours of incubation to become evident. These organisms serve as essential controls to validate the performance and consistency of anaerobic blood agars in supporting the growth and characteristic features of anaerobic bacteria under controlled laboratory conditions. This comprehensive quality control approach ensures the reliability of microbiological testing and enhances the accuracy of diagnostic results in clinical and research settings. Limitations of Brucella agar, not intended for diagnosing disease or conditions in humans. Due to nutritional variations, certain strains may grow poorly or fail to grow on this medium. All presumptive anaerobic organisms must undergo confirmatory testing for identification. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!